welcome to this explainer. Today, we're jumping right into the fundamental chemistry of living things. We are going to completely demystify how this all works, building our understanding from the absolute simplest elements all the way up to the incredibly complex biological machines that literally keep us alive. So let's just dive right in.
Think about this for a second. Does our chemical makeup actually differ all that radically from the Earth itself? Like if we were to take a plant, an animal, or even a microbial paste and compare it to a literal lump of dirt, would the basic chemical ingredients be completely different? Keep that question in the back of your mind as we go.
Here is our roadmap for it today. First, the chemistry of life. Second, analyzing living tissues. Third, micromolecules and metabolites. Fourth, the giant biomolecules. And fifth, the architecture of proteins.
All right, part one, the chemistry of life.
Okay, prepare for a real aha moment. In absolute terms, the Earth's crust and the human body share the exact same elements. Carbon, hydrogen, oxygen. They're in the dirt and they're in us. But, and here's the crucial difference, the relative abundance of carbon and hydrogen is significantly higher in living things than in the Earth's crust. So we are essentially made of the same stardust as the ground we walk on, just arranged in vastly different proportions.
Moving right along to part two, analyzing living tissues.
So how do scientists actually figure this out? How do they isolate these specific chemical compounds within us? Well, it's a surprisingly gritty, literal recipe. Scientists take living tissue, say a piece of vegetable or liver, and grind it up using a mortar and pestle in a chemical called trichloroacetic acid. That creates this really thick slurry. Then they strain that slurry through something as basic as cheesecloth or cotton. This physical separation step is super important because it splits the organic compounds into two very distinct fractions. And that leaves us with a really important split. The stuff that sneaks through the cheesecloth, the filtrate, is called the acid-soluble pool. This has thousands of tiny molecules that were basically just floating around in the cell's fluid. On the flip side, the stuff stuck in the cheesecloth is the retentate, or the acid-insoluble fraction. This part holds the giant structures that physically build the cell itself.
Which brings us to part three, micromolecules and metabolites. Let's zoom in on that acid-soluble pool we just talked about. These are the primary building blocks, and they're tiny, weighing anywhere from 18 to about 800 daltons. These elements arrange into highly functional little bricks. Amino acids, for example, have a central alpha carbon, an amino group, an acidic group, and this really important variable R group. Because that R group changes, we get different amino acids like glycine, alanine, or serine. Alongside simple sugars like glucose and ribose, these are your primary metabolites. They act as the essential fuel and fundamental building blocks for our cells.
But the story gets even cooler when you look at plants, fungi, and microbes. Beyond those basic everyday functions, these organisms produce an incredible array of secondary metabolites. We're talking alkaloids like morphine, essential oils, deadly toxins like ricin, and even polymeric substances like rubber. Now, we don't fully understand the exact role all of these play in the host organisms, though some definitely have ecological importance, but they are absolutely vital to human welfare. They give us drugs, spices, scents, and pigments. It's really wild.
All right, part four, the giant biomacromolecules. Time to scale things up. We're leaving that acid-soluble pool and looking at the giant structures that got caught in the cheesecloth. These biomacromolecules are massive biological polymers, typically weighing 10,000 daltons or more. This includes proteins, nucleic acids, and polysaccharides. Now, there is a quirky exception here, lipids. Lipids are actually really small molecules, under 800 daltons, but they get trapped in this fraction anyway. Why? Because when we grind up the tissue, the cell membranes shatter and form these water-insoluble vesicles, so they get stuck. They aren't strictly macromolecules by weight, but they end up in the macromolecular fraction anyway.
So, what are you mostly made of? Well, water. A whopping 70 to 90% of your cell mass is just water. Following water, proteins take the number 2 spot at 10 to 15%, followed by nucleic acids, carbohydrates, lipids, and just a tiny fraction of ions.
Let's look a bit closer at carbohydrates, specifically polysaccharides. Think of these complex sugars literally like a really long cotton thread built out of single sugar units. Plant cell walls, for instance, are made of cellulose, which is a homopolymer thread of pure glucose. Another variant is starch, which twists into these helical secondary structures. It forms a kind of revolving staircase that brilliantly stores energy for plants. Animals store energy in a super similar way, using a variant called glycogen. And, of course, we absolutely have to talk about nucleic acids. These true macromolecules are polynucleotides, built from 3 distinct chemical components: a heterocyclic nitrogenous base, a monosaccharide sugar, and a phosphate group. Depending on whether that sugar is deoxyribose or ribose, we get DNA or RNA. And as we know, that's the critical genetic material functioning as the blueprint for literally all living organisms on Earth.
And finally, part 5, the architecture of proteins. So, what exactly is a protein? Basically, it's a polypeptide. Think back to those tiny amino acid micromolecules we talked about a minute ago. A protein is just a linear chain of those amino acids linked together by peptide bonds. And because there are 20 different types of essential and non-essential amino acids that can be arranged in any order, proteins are incredibly diverse heteropolymers. That means no 2 protein types are exactly alike. The variety is just massive.
But here's the kicker. A protein thread doesn't just stay as a flat, boring string. It folds. The basic sequence of amino acids is just the primary structure. That thread then folds into a helix or a pleated sheet to form the secondary structure. Then, and this is where it gets crazy, it crumples upon itself, kind of like a hollow woolen ball, to create a highly complex 3-dimensional architecture. That's the tertiary structure. Finally, some proteins actually assemble multiple folded units together, creating a quaternary structure. This folding is not random. It is highly specific and absolutely crucial for the protein to actually do its job.
And why does all this biological origami matter? Because proteins literally do almost everything in your body. This precise 3-dimensional folding makes them the workers of the cell. Collagen provides the intercellular ground substance holding you together. Insulin acts as a critical hormone. Antibodies fight off infectious agents. Receptors are the reason you can smell and taste. It is this sheer structural diversity that literally makes life possible. Now, almost all enzymes are proteins, and these are arguably the most important workers of the bunch. Remember that hollow woolen ball from the tertiary structure? Well, the complex crisscrossing of that protein chain creates these tiny crevices or pockets called active sites. These active sites are the literal engines of life. A substrate fits perfectly into this pocket, allowing the enzyme to catalyze chemical reactions at unbelievably high rates. Without them, the chemical reactions required to just keep you alive would take way, way too long. Which leaves us with a pretty wild thought to chew on. Unlike inorganic catalysts, these intricately folded enzyme pockets are highly sensitive to their environment. They can easily get damaged and lose their shape if your temperature gets too high. So I'll leave you with this. If every essential biological reaction in your entire body depends on the precise, fragile folding of microscopic protein pockets, just how delicately balanced is the chemistry of your life? It's a beautifully complex, yet incredibly vulnerable architecture. Thanks for joining me on this explainer, and keep questioning the amazing world inside you.